Dear students, congratulations on making it to the final lecture. Is on Chapter 20, Part B, Cancer. Cancer is a general term for a whole host of diseases which all have something in common, that being unregulated cell proliferation. Cancer is a term which can only be applied to multicellular creatures, such as animals and plants. As you'll see, there are so many points in the biochemistry of a cell which can go awry that no two cancers in two individuals, or even within the same person, a large cancer, would be the same. There are two types of overgrowth of cells which can occur inside the body. The first type is just called a tumor, where the cells grow out of control, but they are unable to acquire characteristics that allow them to migrate any distance whatsoever. So they stay local. These tumors can become huge, but mostly they're pretty small. Tumors are not normally dangerous unless they grow and block some essential component of the body. And the nice term about tumors is that they are normally benign. That means they don't cause any more distant issues as cancers do. So cancers are also overgrowth of cells. However, in latter stages, cancers can cross barriers. They can spread to other parts of the body. They can be any size, so tiny cancers can kill you, and especially big cancers will kill you. Talking about killing, right now, 1 in 5 humans, 20% of us, will die of a cancer-related illness. And the term that you don't want to hear with a cancer is malignant. A malignant cancer is one that is spreading. Cancerous cells either have or will acquire the ability to invade other tissues within the body. So by definition, cancer is an invasive disease. It's not easy for cells to acquire the necessary components to become invasive, but once they do, they're pretty difficult to stop. One of the early characteristics of a cancerous mass is its ability to break through barriers such as the basal lamina. Once they do that, the cells are said to be metastatic. And being metastatic simply means invading other tissues where those cells were not destined to be. Once the cells acquire further abilities, such as the ability to live inside lymph or blood and establish themselves in more distant tissues, then colonies can be established. So this is called the primary colony, where the cancer first arose, and any other sites that this same cancer finds itself growing will be called secondary growths. This figure here shows a nice example of secondary tumors established in this patient's liver. The cause of death in this case would be the non-functioning of a normal liver. So the liver cells that make up the healthy liver have been overtaken and crowded out by the presence of the tumors themselves. So lack of function of normal liver has killed this patient. An alternative physiological cause of death from cancer is the release of unnecessary substances. So if cancer cells are releasing a hormone, then the number of cancer cells is far in excess of normal, and that hormone that's been released will be overwhelming the body and upsetting biochemical ratio. Whereas normal tissue is highly organized, as we found in the first part of chapter 20, tumors in general are disorganized, and their primary purpose is to grow out of control, and they don't worry about their organization at all. That contrast is clearly indicated if you look carefully in the top right-hand corner. That's the mass, the tumor, and below that is the organized healthy liver of the patient. We now have a much better grasp on what causes cancer, even though we don't have the full story. Epidemiological studies where we compare different populations against other populations reveal that environmental factors play a large role in the development of disease. In particular, lots of people cite the early Japanese studies of Japanese migrants moving first from Japan to Hawaii, and then their children growing up on the mainland here in the United States. These suggest that where you live currently is more important to your outlook on developing cancer than where you were born. Most people are aware that lifestyle is very, very important to your risk of developing cancer. So smoking, heavy drinking, and obesity are known factors that increase the rate at which cancers can develop in that individual. But also viruses, especially retroviruses, can be responsible for many cancers. And cervical cancer is the one that's in the news quite recently due to the easy spread of human papilloma virus. Cancer is a result of genetic damage within cells. Current data suggests that multiple genes have to become mutated or defective before a malignancy can develop. Some scientists believe that the number is close to 10. 10 different genes need to be knocked off individually over time before a cancer can develop. However, these mutations are restricted to certain classes of genes that are involved in the cell cycle, DNA repair, and propagation. Thus, it makes sense to view cancer as an age-related disease because it takes time for these mutations to accumulate slowly but surely over the life of the organism. Once damaged cells begin to accumulate in the body, they undergo more damage. So it's a self-fulfilling prophecy. And that leads to greater and greater genetic instability as the number of cells begins to increase. Since most animals are made up of somatic cells rather than germ cells, it's quite common that those somatic cells are the ones that are normally going to lead to cancer, due simply to their overwhelming numbers. So cancer is fundamentally a genetic disease, but it's mostly a disease of somatic cells rather than germline cells. For this primary reason, cancer is a disease of an organism rather than a disease of inheritance. Although the progeny of the initial cancer cells do inherit it through mitotic cell divisions into daughter cells. The assault on the chemistry of a cell can take place from outside the cell or from within the cell. Either way, you cannot prevent this type of chemical instability, nor can you shut down the general mechanisms of cell biology such as DNA repair, DNA replication. So errors are going to happen and they can't be prevented. But if those errors are increased, then the chances of developing cancer proportionately increase. The reading of the chapter presents some fantastic mathematical evidence based on sheer numbers. We know from a previous chapter, chapter 6, that DNA polymerase and DNA repair mechanisms try to be as faithful as possible when it comes to replicating and repairing DNA. But even after all those processes have done their jobs, mistakes are left behind. Approximately one mistake for every billion to 10 billion nucleotides copied. That may not seem much, but in context, it's quite large. Any single gene out of the 21,000 that we do have has a 1 in a million or 1 in 10 million chance of being hit by a mutation at every cell division that cell performs. So some cells are dividing more than others, so those cells are more susceptible to becoming cancerous. A human being generates 10 to the 16 number of cells in their lifetime. So that's replacement cells. That's a huge number. So mixing these numbers together, you find that on average, individual genes can become damaged about 1 billion times in their lifetime. That is amazing. So the question is not why do we don't develop cancer as multicellular organisms? The question is why don't we develop more frequent cancers? And we have to thank the immune system for its surveillance and its dedication to eradicating many, many cancers that develop each and every day. A quick chromosomal spread taken from a breast cancer cell reveals that chromosomal mutations are quite common. So mutations of chromosomes can occur at the general level, and we see that taking place in this figure. Many chromosomes have transposed themselves onto the ends of other chromosomes, and some chromosomes have gone through reciprocal exchange of DNA. Other parts are missing and other parts are duplicated. So in this spread, we have lots of DNA damage. It's damage such as this which accelerates the changes in the phenotype of the population of cancer cells within the body. As we mentioned, the more cells you have in your body, the more likely that different populations of those cells will have different types of genetic damage, therefore making treating a large tumor more difficult than treating a small tumor on average. Under normal circumstances, each cell in the body is kept in check, and if it starts misbehaving, it then either dies or is killed by the immune system. But those apoptotic pathways can be disabled by cancer cells as one of their mutations. So precancerous cells and cancer cells can acquire defects in their cell cycles and in their DNA repair cycles and in their apoptotic pathways, therefore giving them this ability to outrun these safeguards. And as they do so, the mutations in the cell cycles allow the cancer cells not to wait for the quality control steps to be implemented, but to proceed to the next stage regardless, therefore not only growing faster and faster, but sustaining more and more damage in the process. So the population of cells inside an organism can be looked at as a population of individuals in a species. So from the same perspective, evolution can work on both sets, i.e. the number of elephants in a herd versus the number of cells inside the individual elephant. And advantage can be gained by doing something that your neighbors can't do. So in this panel here, we can see through mutation, one cell has acquired a phenotype that its neighbors don't have, and that may be to grow faster than its neighbors do. So as the cell produces more and more progeny, in this case 5 cells, more mutations will accumulate. And maybe one of those daughter cells will accumulate a second mutation that will give it a second advantage over its own cousins. Now that cell will detach itself from the basal lamina and be able to form a mound, and all the cells in that mound will be descendants of this one cell. And over time, another mutation will appear in this population of cells, and that's indicated by the red cell. And that red cell will acquire another feature, another phenotype that the pink cells didn't have. Maybe the ability to melt the basal lamina and invade the tissue below. And maybe another cell in this population, not shown, will acquire another ability to invade the blood or the lymphatic system. And it goes on from there. But this acceleration in genetic defects not only accelerates, but leads to a more devastating outcome. In summary, as the number of descendants of the original cell increases, more and more changes will be experienced by the progeny of those cells, some going in one direction, others randomly going in a different direction, depending on chance events. So the book talks about epithelial cells that become malignant in the previous figure, and they show that on average, this is a general mechanism by which the changes will accumulate, allowing those cells to then become metastatic. So take a few seconds and make sure that you can relate this back to the figure on the previous slide. We mentioned previously that not all genes mutated lead to cancer. So which genes do? Well, there's a set of genes, about 5 sets, that are very important. 2 of those 5 sets are mentioned here, oncogenes and tumor suppressor genes. So let's look at those for a few minutes. So tumor suppressor genes are genes which encode proteins, and those proteins are evolved to monitor and watch over cell functions that are very important. And they act as policemen inside a cell. Their job is only to permit the cell to transition to the next phase in its biochemistry if all conditions are met, and to put the brakes on should there be any kind of issue. They can halt the cell cycle activity when necessary. So they can interact with the cyclins and prevent the cyclins from upsurging the cell into the next stage of the cell cycle. These type of mutations require both genes to become defective. So there has to be a loss of function. So these proteins that we mentioned earlier, they have to be knocked off, i.e. inactivated. So those functions have to be removed before a cell can even consider becoming cancerous. Because a mutation in one of the two genes is not enough in itself to cause the phenotype, we call these recessive mutations. Both genes have to be mutated before the phenotype of the cell can change. On the contrary, another set of genes called oncogenes, they exist, and their normal versions are called proto-oncogenes. So these are the normal ones that we find inside the cell. And they're normally part of the cell's machinery in driving the cell cycle or DNA repair or cell division. They only require one copy to become defective. That one copy can then produce an abnormal product, which is in itself enough to upset the balance within the cell. So those are called gain of function mutations. And because only one copy is necessary to become defective, those are therefore dominant mutations. The normal copy cannot prevent this activity. This slide takes a slightly deeper look at the same principle. So in this particular cell, could be one cell in the body, a somatic cell or a germline cell, and that cell is normally protected by two alleles of the same gene. And if those two alleles are normal, they'll produce two sets of normal protein. Even though one set is enough to protect the cell has a safety net of producing two. And as we can see here, mutation at some point in the life of that cell or its offspring can inactivate one of these two genes depending on environmental conditions. And if one copy is inactivated, there's no effect on the cell because the other copy, the other allele, keeps everything nice and healthy. However, if a second event takes place, which accidentally knocks off the remaining good copy, now you have two bad copies, and neither one is available to protect the cell, and now the cell will do whatever those genes were trying to prevent from happening, i.e. maybe it won't be able to repair its DNA properly or do something like that. So this is how the system here works. The same type of analysis performed on proto-oncogenes, as we see in this cell, it has two normal versions, suggests that all it takes is a mutation to hit one of these genes that then supercharges that gene in some way, therefore there's a gain of function. This slide here shows some of the ways in which those activities can be supercharged. So here's your normal gene, the proto-oncogene, and through mutation, you can then change the protein that that gene produces. In this case, making the protein overly active. For instance, it could be a protein in the cell cycle, like a cyclin, and that cyclin is now very aggressive in adding phosphates to other molecules. Alternatively, a proto-oncogene can be turned into an oncogene where its products are produced at a much higher rate, either through gene duplication or gene activation. Regardless, the normal protein is now overproduced, and each copy magnifies the functionality, just like we saw here. And for the sake of completeness, a third example is given where proto-oncogenes can be converted to oncogenes by chromosomal rearrangements. So chromosomal rearrangements come in many different flavors, but the point is that they upset the normal biochemistry of cell-protein interactions. This slide addresses the same principle with loss of function genes. One pathway to bad chemistry is to be given by one of your parents a broken gene in the first place. So the only copy you have came from your other parent, and hopefully that second copy will prevent that cell from acquiring further damage and keeping that cell healthy. Alternatively, we have other pathways too. A chromosome can be lost through non-disjunction during any type of cell division, meiosis or mitosis. And that only leaves you with one copy, and if that's the copy that's damaged, then this cell is no longer protected. An alternative strategy is rather than losing the whole chromosome, you may just lose the regulatory part or the gene itself, and that has the same outcome. Alternatively, you could have a mutation that directly nullifies the protein made by the second copy. Now you have no protection whatsoever from either allele. And finally, the normal copy can be shut down by epigenetic modification of the histones or the DNA backbone by accident or by design, such as viruses. In all 5 cases, you end up with a heightened risk of developing cancers. Once we compile all the data that has been amassed on how cancers develop and the underlying biochemical changes taking place in cells, we get this very unified picture of 3 general processes which have to break down before cancer can become a problem. The first of those pathways has to do with cell proliferation. The second pathway has to do with DNA duplication and DNA damage management. And the third pathway has to do with cell growth. So this is increasing numbers. This is making sure that your DNA is copied properly and segregated to the 2 chromosomes. And this has to do with an increase in the size of the cell itself by acquiring more chemistry. For a real malignancy to develop, all 3 pathways must be broken at some point during the history of that line of cells. It can happen slowly, one at a time, or it can happen slowly in some cases and faster in other cases. Regardless, in most cells that are examined to be malignant, all 3 pathways are defective. Now, these pathways can be quite complex. They can involve tens to hundreds and even thousands of genes. Any one of those genes or proteins could be broken along the pathway. So in one person, gene number 6 in this pathway could be broken, and gene number 19 in this pathway, and gene number 422 in this pathway. So no 2 cancers necessarily involve the same genes being broken. Therefore, each cancer may be unique to that individual. One category of cancer where we have lots of data is colon cancer. Colon cancer results normally from the development of tumors known as polyps. And those polyps are present on the inner surface of your digestive tract, either in your small or normally in your large intestine. A common finding is that in most tumors of the colon, a particular protein is defective. And that protein is a tumor suppressor gene known as APV, which stands for adenomatosis polyposis coli, APC. Tissue sections taken from these patients show that both alleles, both copies of the gene in those malignant cells are damaged. Whereas in healthy tissue taken from the same patients, at least one of the 2 alleles is working. Even with that said, if we go back to the original statement that cancer is a multi-mutation theory, then we should see that happening inside colorectal cancers. And we do, and that's illustrated here. So the loss of the APC locus results in a change in phenotype of the cells. So they form these polyps. But the polyps themselves aren't cancerous yet. But as their numbers increase, then inside some of those polyps, a second mutation will take place. For instance, in the gene for RAS. RAS is one of those regulatory proteins that's looking after the cell cycle. But even that is not enough. You just get a small tumor, a localized tumor, that is not able to spread to other tissue unless another tumor suppressor gene, one of the other hundreds, is mutated. But even then, the tumor is still localized, but it's getting larger. And maybe another mutation in the P53 gene, another tumor suppressor gene, has taken place. So now the tumor becomes invasive, but it may still not have acquired the ability to grow somewhere else in the body, like the liver or the brain. Eventually, those mutations will be acquired as the size of the tumor increases. The likelihood of this happening increases. And some of those cells, some of those progeny cells, eventually will gain the ability to grow and live elsewhere. And that's the danger of this type of cancer. Early checkups for bumps and lumps are recommended at all times because numbers are against the patient. The more cells that are aberrant, the more likelihood that they themselves will multiply faster. And the faster they multiply, the more likelihood that they'll gain more and more genetic abnormalities. So the smaller the mass, the better the prognosis. Here you can see in a logarithmic scale the number of cells present in tumors of different sizes. So a tumor that's 0.1 millimeter in diameter cannot be detected, and that will contain a very low number of cells. Whereas a tumor at 1 centimeter, the standard for classifying stage 2 cancer, would be here. That would contain about 10 billion cells. So when you feel a lump somewhere in your body and the lump is about 1 centimeter, you already have a problem. There's a billion cells or 10 billion cells at that location, and some of them may be more aggressive than others. But as the size of the tumor increases, then the likelihood of developing something abnormal within those masses also increases. So that's indicated quite nicely in statistical data as presented here. The final pages of the chapter, they talk about therapies utilized to treat patients with cancers. So the standard has been in the past chemotherapy. Chemotherapy is a poison which penetrates the entire body, and its job is to interfere with cancer cells more than the normal cells of the patient. So each chemotherapy drug is looking for a weakness in the patient's cancer that can be exploited during the treatment phase, so killing cancer cells rather than killing the patient. Another treatment option is radiotherapy, where X-rays are used to damage the DNA of cells close to and within the tumors. A third methodology is to surgically remove the tumors when found, but sometimes the tumors may be too diverse or too small or in strategic positions in the body that may endanger the patient's life. More current treatment options stem from cell biology research. If a cancer has an abnormal enzyme or it has a missing component, one solution is to treat that patient by deactivating the abnormal enzyme or by replacing the missing component. And this is what this slide is showing us. So one company developed a substance called Gleevec. This drug is simply an interfering molecule which binds to and blocks the active site of a hyperactive kinase, ABL. So the enzyme is now dumbed down again to low levels, therefore shutting down the high activity of those cancer cells and bringing them back into compliance. More powerful stories are emerging constantly as scientists discover the inborn potential of the immune system to be trained to seek and destroy a patient's cancer cells. So in these therapies, the tumor is exposed to the white blood cells of the same patient in the test tube, and the scientist filters out those cells that are going to be very aggressive in identifying and then destroying the cancer cells. These so-called immunotherapies are the most positive result in the last few years when it comes to treating cancer. Thank you.